So good morning students. So I am Dr. D. Gavaskar, Associate Professor, Wales Institute of Science, Technology, Advanced Studies, and this semester we will be discussing about the phytochemistry practicals. So what is the introduction of this phytochemistry practicals? So we are going to extract only the secondary metabolites. So what is maybe secondary metabolites? So if you take phytochemicals, those chemicals will be found in plants and we will be extracting those chemicals. And these chemicals will be classified into two types. One is primary metabolites and secondary metabolites. So primary metabolites, which is essential for the metabolism and plants growth. For example, carbohydrates and proteins, these two compounds are coming in the category of primary metabolites. So if you take secondary metabolites, flavonoids and terpenes and alkaloids, these compounds are called as secondary metabolites, and these compounds, it will form from the primary metabolites for the safety purpose. And for example, I can say curcumin is secondary metabolites, is a flavonoids compounds, and this pectin is a polysaccharides, and nicotine is a secondary metabolites, and that beta carotene is a secondary metabolites. So these are some secondary metabolites, and we will be discussing some experiment, maximum 8 experiments.
This is our first practical, that is the practical is isolation of pectin from orange peel. Yes. So this is the structure of pectin. If you happen to look at this structure chemically contains, that is a polymer of uronic acid and the methyl ester. If you happen to look at this molecule, you have carboxylic acid functional group. So this part is said to be uronic acid, and it is having the methyl ester functional group. This is the methyl ester of uronic acid. So it is a complex structure. I have written here. So this is a complex polysaccharide which is containing basically what? D-galactouronic acid derivatives. So this is a structure of pectin. So this is what we are going to isolate from the orange peel. Fine. So this, if you happen to look at this molecule is generally found in middle lamella of plant cells. For example, what are the fruits that we can isolate is, that is, you can isolate generally from orange peel, and this is lemon peel, and you can isolate from sunflower head and raw papaya. From these species, you can isolate this molecule. Fine. So this molecule was first isolated by Henry Braconnet in the year of 1825. So with this basic introduction, so let us start the practice.
So how we can extract the pectin from orange peel? So if you see this orange peel, we have the white layer, right? So this white layer is called as pectin. Okay. So already Sir told it's made up of the polysaccharides compounds. And what is the application of this pectin? It contains two important applications in food industry. It is used as a gelling agent. Okay, and it will act as a additive. So in order to make the food as thick or viscous compound, they will add this. Okay, and if you see the pharmaceutical application of this pectin, it is used to reduce the cholesterol in the intestine, and is a dietary supplement. So if you have constipation, means it will able to release the constipation because it's contained full of fibers. So this pectin compound, which is present 30% in orange peel. Okay. So how we can extract? First you have to take this orange peel, okay, into different parts, and take 10 gram of pectins. That's orange peel. Just take 10 gram of orange peel and add 100 ml of distilled water. Okay, take a beaker, take 100 ml of distilled water, take 10 gram of this orange peel and eat that solution for 3 hours. So before that you have to add hydrochloric acid or citric acid. You have to maintain the pH, acidic pH. Okay, take this pH paper, add concentrated hydrochloric acid, dropwise, check the pH, you know what's the color. Okay, if you see here, this pink color is a indication of acidic. Okay, you have to maintain the pH approximately 4.5. Okay, 100 ml of beaker, add water, add orange peel, add dilute hydrochloric acid or concentrated hydrochloric acid, check the pH. If it's acid, heat the solution for 3 hours. Okay, so after 3 hours, take the solution and cool the solution. Take nylon cloth, filter, remove the dust particles, waste particles, take the filtrate, and you add isopropyl alcohol or N-propyl alcohol. Just add approximately 50 ml. Just add. You will get the jelly compound which is precipitated. So it contains full of water. Okay, just filter the jelly compound for 1 hour because it releases lot of waters. After 1 hour, you will be able to see the white color paper like layer that is called as pectin. Take the weight, note down in the observation note. Okay, this is the entire procedure. If any doubt, you may ask. See, actually, why are we heating the solution? Because then only this cell wall will be smoothened. Okay, that's why we heat. And you have to add acid because in that particular pH only it will precipitate it. So these two things is very important. Okay, shall we start the experiment? So we are going to extract this pectin from orange peel. So what is the requirement for this experiment? So we need 10 grams or 50 grams of orange peel. Just add this to the beaker and then add approximately 300 ml of distilled water. Then only it will initiate that loosing process. You have to make that layer softer. Okay, and then so this is a 3N hydrochloric acid solution. So I am going to acidify this solution. So it will increase that smoothen process. Okay, add this hydrochloric acid dropwise. Glass rod, gently, and check the pH. Now also is neutral. So I am going to add some more amount of this hydrochloric acid. So you have to add the solution until the solution will be acidic. Just swirl it and then check the pH. So now it's slightly acidic. Okay, so this pH is enough. Are able to see that pink color? So it shows that this solution is a acidic solution. You just stop adding this hydrochloric acid and then heat this solution through Bunsen burner, and you have to heat it this solution for 2 hours. So where we are heating this simple process to remove that cell wall, because that compound is present in the lamella of the cell wall of the orange peel. So after 3 hours of heating that orange peel, the substance, it will look like this. If you see the solution, all the peels was dissolved into the water, and all the things, it was extracted into the aqueous solution. So now you can stop this heating process, and you have to wait for 5 to 10 minutes, because then only this all the substance, it will be digested, and the temperature should be in the room temperature. So after that, you have to take this solution and you have to filter to the nylon cloth. So after filtered, you have to take that extract, and then you add sufficient amount of N-propyl alcohol. So obviously that pectin will be precipitated. I am going to filter this solution. So with help of this filtration, I can remove the dust particle. You have to take only this filtrate. Use only that nylon cloth instead of that Wattman filter paper or ordinary filter paper. Just use this nylon cloth. So this orange contains 30% of this pectin, which is used as a gelling agent and then food additive, and it is used to reduce the cholesterol in the intestine, and it contains a fiber. So this is very useful for the relief from constipation. And this citrus food contain vitamin C, antioxidant. I will take this citrus food regularly. It will increase the immune of the human being. Okay. So you can use isopropyl alcohol or ethyl alcohol or N-propyl alcohol for the precipitation of the pectin. So this pectin is a derivative of galactose. Derivative of galactose. The galactose is the epimer of dash saccharide. Any idea? So what is epimer? So now we are doing the filtration process. So this is a extract. So this is a aqueous extract. The compound is highly soluble in water. So just take this and do like this. Just do like this carefully, because very hot. So you have to do carefully. So now you will check the pH of the solution. We will check the pH of the solution. Glass rod, glass rod. So now is it's a neutral, it's a neutral solution. Okay, so previously it was acidic, now it was neutral. After that, just take N propyl alcohol. So here I am taking sufficient amount of N propyl alcohol. Could you able to see that jelly compound which is precipitating out? So once again I am taking some amount of N propyl alcohol and just adding it. Come here, just see. Be able to see that precipitate. Okay. So wait for another 5 minutes and then filter. Take the weight. Take down in the observation notebook. Okay, is clear? Any doubt? Okay. So filter, dry it, and note down the weight. Now take down the observation notebook. The obtained yield of the pectin. Okay, so drying should be important because this compound is a deleterious substance. Dry it properly and then take weight. So we got the white gelatinous compound that is called as pectin. So use this nylon cloth for the filtration process and I am going to filter this. You add gently, don't spill the solution. Okay. So you can use lemon also for extracting this. Lemon which contain 50 percentage of this pectin. Okay, and apple 0.5 percentage apple, and Mozambique it contains 15 percentage of this pectin. Orange and lemon. Lemon is which contain more quantity of this pectin. So if you see this, the compound is precipitating out. Okay, so you have to convert this hot solution into normal cold solution. Be able to see. Rod, glass rod, glass rod. Do like this gently, okay? So you have to remove this water. That's why I'm using this nylon cloth. So once again, add this extract. So like this, repeat this process until you have to get sufficient amount of pectin. Be able to see that white colored solid, that is pectin. Make the filter paper that not to wait that pectin. I need filter paper, ordinary filter paper. So remove all the compound with the help of this glass rod. So now we are going to put the weight. Okay. So you have to dry this and you have to put the weight. Okay. Get it out.